Hi everyone, welcome to Dento'ZEN. We are continuing with our lecture on morphology and physiology of bacteria. The topic is divided into classification, types of microscopes, staining, shapes, anatomy of bacteria and their growth and multiplication. We have already discussed classification and types of microscopes in the previous video. You can check it there. We are going to discuss staining methods today, which can be gram staining, Ziehl staining, which can be important short notes for your exam, and the different shapes of bacteria as seen under microscope. Remaining two will take up in next video. So let's start. Before starting, quickly subscribe to Dento'ZEN, tap on the like button and hit the bell icon so that you remain notified about new videos. Now, why staining is important? If we try to look at the live bacteria under light microscope, they will appear something like this. That is, we cannot see their structural details. But when we stain them, there is a color contrast and there is increase in their visibility. So stained section bacteria will appear like this. Now, when we stain live bacteria, that type of staining is called vital staining. That can be your important viva entrance question. That means cells remain alive even after staining. Now, but most routine procedures, we have to kill the cell, kill the bacteria. So that is called fixation of bacteria. So now, how is that done? So first we'll prepare a smear. What is a smear? We take a glass slide, and from the specimen that we have collected from the patient, which can be blood, urine, saliva. So from that specimen, we put a thin layer, prepare a thin layer over this glass slide, spread a thin layer of that specimen over the slide, and now this is called bacterial smear or bacterial film, and now it is dried in there and then it is fixed. So drying and fixation of smears to kill the bacteria. So why fixation is done? It protects the internal structures so that we can see them clearly, and it prevents the movement during staining. So that is why fixation is important. We kill the bacteria. How it is done? There are two methods. First is heat fixation. We bring the heat, we pass this smear gently over the flame, that will fix the bacteria to the slide. And second method is methanol fixation, where we use methanol to fix the smear, specially used for blood smears where our specimen is from the blood. So now we have prepared our smear, we have fixed our smear. Now we are going to do the staining. Let's see the common staining techniques. First is simple stains. Basic dyes are used like methylene blue or basic fuchsin. So they will give a color contrast to bacteria, but all bacteria will take up same color as we can see here. So that is simple stains. Then we have negative staining, which is important short note. So what is happening here? We mix the bacteria with the dyes. Dye that is used is India ink or Neelsen. So this will give black color to the background, but bacteria or their structures like capsule will remain unstained. So here, as we can see, this unstained capsule can be seen against the dark colored background. This technique can also be used for slender bacteria, very thin bacteria like spirochetes, where unstained bacteria can be seen. So these are the spirochetes. So that is negative staining and important short note. Now third is impregnation methods. Now some structures or cells are too thin to be seen under microscope. How do we see them? Then we impregnate them with the silver salts over their surface. We deposit these salts which make them thicker, and now they become visible. Here comes the viva question. Where do we use these techniques? To demonstrate flagella, bacterial flagella first, and second is again to see thin bacteria like spirochetes. Fourth is the differential stains. Here two stains are used. They will give different colors to different bacteria. So let's see the example. First is gram staining. Here the bacteria are divided as gram positive or gram negative based on the color they take up. Second is acid fast staining, where bacteria is divided as acid fast or non-acid fast, again based on the color they take up. And third is Albert stain, where the bacteria are divided as the ones which contain metachromatic granules or which do not contain. So these three are examples of differential staining. Let's talk about them one by one. First important is gram staining, which can be important short note or viva question. So it was originally developed by Hans Christian Gram, and that is why the name gram staining. It is still the most widely used method in the labs for diagnosing bacteria. According to this method, bacteria can be gram positive, which take up violet color, which appear violet under microscope, or they can be gram negative, which appear pink under the microscope. Now, why this type of two bacteria are there? Let's see the technique. So here, as we said, it is a differential stain. So two stains are there. So in the technique, the first step is first stain will be put over this smear that we have prepared. So the first stain is primary stain, which can be crystal violet or methyl violet or gentian violet. It is kept for 1 minute and then it is rinsed. What will happen now? This stain will be taken up by bacteria and now they will appear violet. But this stain cannot remain inside bacteria for a long time. So how do we increase the time? So that is done by the second step. We bring the mordant. So what is the role of mordant? The mordant that is used is grams iodine. That can be viva questions. So grams iodine is kept over it 1 minute. This iodine will now bind to the dye, that is gentian violet, and will form a complex, iodine dye complex, which will help in retaining this dye, retaining this stain inside the bacteria for a long time, so they can remain violet for a long time. Now comes the third step, that is decolorizer. What is the role of decolorizer? D means removal, so color removing agent. So the decolorizer that is used is ethanol, acetone or aniline. After this decolorizer is put, then it is rinsed. So in some bacteria will still keep the first stain, that is they will still be violet, but in some bacteria the first stain will get removed by the decolorizer and now they will again become unstained. So how do we see these unstained bacteria now? Here comes the role of our second stain, that is counter stain. So now we'll bring the counter stain in the fourth step, which can be carbon fuchsin, safranin or neutral red, that is put for 30 seconds. After that, the bacteria which lost their color in the third step now will take up the counter stain and now will appear pink. So that is how the two types of bacteria can be seen: gram positive, which have retained their first color, they will be violet, and gram negative, which have got second stain, counter stain, and now they appear pink. So that is the interpretation. That is, if we look at this gram stain slide under the microscope, we can see two types of staining. That is, violet colored will be the gram positive bacteria and pink will be gram negative bacteria. Bacteria can be of different shapes. They can be round, they can be rod shaped. So gram positive are those which resist decolorization. They do not get decolorized. They retain the primary stain, so they are still violet. But gram negative, which were weaker, they got decolorized, their first stain got removed, so they had to take up second stain, counter stain, so they appear pink. So that is the interpretation. So why it is important to divide bacteria as gram positive or gram negative? Because not only they are different in their colors, but also they are different in their characteristics, other properties. So why this happens? Why some bacteria are positive, why some negative? That is the principle of gram staining, which can be important viva question or short note. Let's see the gram positive bacteria and gram negative. So there are two important reasons. First is, if we look at the protoplasm of the gram positive bacteria, they have highly acidic, more acidic protoplasm as compared to negative, and this acidic protoplasm can keep the basic dye, iodine dye complex, for longer time. So more acidic protoplasm. Second reason is cell wall of these bacteria, gram positive, if you see, the cell wall is thicker as compared to gram negative. So thick peptidoglycan layer, about 50 to 100 layers are there. That is the very important reason that forms a permeability barrier, strong barrier, which do not let the first stain, that is crystal violet, to come out. So they still remain the violet stain and they appear violet. Whereas gram negative bacteria, this wall is very thin. It has thin peptidoglycan layer. So first stain will come out and now they have to take up the second stain, that is they will appear pink. So they have more permeable wall, and also they have lipopolysaccharides which will easily get disrupted by decolorizer. So first stain will come out and they will have to take up second stain and appear pink. So those two reasons, more acidic protoplasm of gram positive and thicker peptidoglycan layer in the cell wall of gram positive. Now let's talk about second, acid fast staining. This was discovered by Paul Ehrlich and modified by Ziehl and Neelsen. So some bacteria which have mycolic acid in their cell wall, they are called acid fast bacilli, whereas the others are non-acid fast. So let's see the technique. Again, there are two stains. So first we prepare the smear. Now these acid fast stain bacilli, example is Mycobacterium tuberculosis. So this, for these patients who have tuberculosis, sputum is the sample. So we take the specimen, sputum specimen, and from there we prepare the smear. We take the yellow part of the sputum, yellow purulent part of the sputum, and then it is spread over the glass slide. Then it is air dried for 15 to 30 minutes and heat fixed for 3 to 5 times for 3 to 4 seconds. So that preparation of smear is important. After that, we do the staining technique in which we will have two stains. So let's see, in the first step we'll bring our primary stain. Now the primary stain that is used here is carbon fuchsin, which is kept for 5 minutes. After that, we do intermittent heating of the smear, that is we pass it over the flame intermittently till the vapors rise. So bacteria will take up the primary stain, carbon fuchsin, acid fast bacilli. Second step again is decolorizer, that is now we want to remove the color from the bacteria which are not acid fast. So how do we do that? The decolorizer that is used is 20% sulfuric acid. That can be important viva question. Now it is kept for 2 to 4 minutes. So non-acid fast bacilli, the color will get removed and now they will be unstained. whereas acid fast bacilli will still remain their first color. So how do we see these unstained? Now we bring the second stain, that is counter stain, which is 0.1% methylene blue, which is kept for 30 seconds. Now these unstained bacteria will take up this blue color. So here we can see two types of bacteria. The one which retained the primary stain, carbonyl fusion, they appear red, and the ones in which color got removed in the second step and they have taken up the blue color, so they appear blue. So interpretation, when we see this stain slide under the microscope, we can see like this. So our acid fast bacilli, example is Mycobacterium tuberculi. They will appear long, slender, thin bacteria, straight or slightly curved. As you can see, these are the acid fast bacilli, slender, slightly curved or straight or beaded, and they will be red. So whereas non-acid fast bacilli, as you can see in the background, they will be blue in color. So that is non-acid fast blue color. Now this staining can also be used for spores. So that is important point here. Now examples of acid fast bacteria are Mycobacterium tuberculosis and Mycobacterium leprae. So for Mycobacterium tuberculosis, 20% sulfuric acid is used as decolorizer. For Mycobacterium leprae, we can use 5% sulfuric acid, but we have to use sulfuric acid. Remember that can be important viva question. Now we come to the Albert stain, that is used to see metachromatic granules of Corynebacterium diphtheriae. So the technique is, again we have two stains. First step will prepare the smear, heat fix it. Second step, first stain will come, that is Albert 1 or Albert stain, which is put for 5 minutes. So this stain will contain toluidine blue, malachite green, glacial acetic acid, alcohol and distilled water. After that we remove it, we drain the solution, and then we bring the second stain, that is Albert 2 or iodine solution, that is kept for 1 minute. It contains iodine in potassium iodide. After that we wash it with water. And now we look at the slide under the microscope. What is the interpretation? So our Corynebacterium diphtheriae, they will appear green colored bacilli, as we can see here. So these are the Corynebacterium diphtheriae in Chinese letter or uniform pattern, and at their ends, if you can see, these black blue are the metachromatic granules at their polar ends. So that is how we identify Corynebacterium diphtheriae. And the others, diphtheroids, which do not have this metachromatic granules, they are arranged in palisade pattern. So that is the Albert stain for Corynebacterium diphtheriae. Now we talk about different shapes of bacteria. So bacteria under microscope not only stain differently but can also have different shapes. They can be cocci, coccus means berry, they can be spherical or oval shape. Bacilli, baculus meaning rod, so they can be rod shaped bacilli, that is bacilli. Then third can be vibrio, that is comma shaped or curved. So they have a vibratory motility, so they are vibrios. Then we have spirilla. Spirilla are rigid spiral forms. Spirochetes, spiro referring to their coiling, and chete meaning hairs. Then actinomycetes, they are branching filamentous bacteria, so those are actinomycetes. And mycoplasmas, they do not possess stable shape. They can be oval, they can be round, they can be or interlacing filaments. So those can be the different shapes of bacteria. Now how are they arranged? So they can have different arrangements, like they can be in pairs in 2, or they can be in chains, they can be in groups of 4, groups of 8, or they can be in grape-like clusters. So both cocci or bacilli forms can be shown in different arrangements. Also they can be gram positive or gram negative. So these are the different arrangements. Now also there can be vibrio, spirilla, spirochetes. They can also be in groups like this. Now that's all. Let's check what have you learned. So example of negative staining: gram staining, acid fast, Albert staining, India ink preparation. Which one is negative staining? Second is impregnation method is used for flagella, spirochetes, both or none. Third is gram positive bacteria have what? Acidic protoplasm, thicker peptidoglycan in cell wall, both or none. And decolorizer that is used for acid fast staining or Ziehl staining is nitric acid, sulfuric acid, carbonyl fusion or methylene blue. Now I am giving you some pictures and you can identify the type of staining. First, A. So here all the bacteria are same colored. So what type of staining is that? Second is we can see unstained bacteria or their capsules against a dark background. What type of staining is that? Third, we can see two types of stains, that is violet and pink colored bacteria. And then third is we can again see rod shaped bacilli bacteria which are curved, which are pink, red in color with blue in background. And the last staining where we can see green curved bacteria with dark black granules at their ends. So let me know about your answers in the comment section below. So that is all for this video. If you really enjoyed the video, do tap on the like button and share this video with your friends. Keep watching, keep learning, keep smiling. See you in the next video soon. Till then, take care. Bye bye.